and welcome to a new episode of the Computomics podcast. Our guest today heads the Bioinformatics Research Group at TUM Campus Straubing and University of Applied Sciences Weihenstephan-Triesdorf, and he's a two-time winner of the Prize for Excellence in Teaching. Welcome, Dominik Grimm.
Thanks, and thanks for the nice introduction.
You're welcome. It's great to have you here. We have a few topics we'd like to talk about today, but I'm curious, kind of as an icebreaker, what's your favorite tree?
My favorite tree? Oh, that's a very good question. Apple tree, maybe?
Why?
Because it gives you fruits. It's a tree, and so you can also use it in daily life. Yeah.
I asked you again. It's a bit of an icebreaker, but you received a tree as the actual prize for excellence in teaching. So, would you say? What are values that you have with regard to teaching in your role at TUM?
Yes, so teaching is very important for myself because I think it's something where we can educate our next generations of students and we can give them something that's important for the future. And I like this prize of a tree a lot because the tree is something special. It will be there for forever, more or less, or for the next few years, few hundred years, maybe not an apple tree, but other trees will be there quite a long time. So it's something sustainable. And that's also something that we do very much at our campus. We concentrate our research on sustainability, and we have to somehow go into a new world where we are more sustainable. And that's why I like this tree a lot, because that's something sustainable that will be there for several years. And it's not just a toy or something that you can hang on your wall and that will disappear. The tree will be there.
Mm-hmm. True. So it's a prize for you, but also almost a gift to the community there and the overall community in a way, very sustainable. And maybe something we should copy with other prizes.
Yes, I like the idea.
Yeah, so you already mentioned sustainability is a huge part of what you do. What would you say is your focus area in your work at TUM?
Yeah, so we focus on mainly developing new statistical models and machine learning models, but that we can apply them on real-world application, and especially to agricultural and biological applications, but also more and more into the fields of chemical process engineering, for example. So we try to enable with the research we are doing that scientists are able to bring our methodology towards applications, more or less. And that's also very interesting at our campus here, that we are very interdisciplinary, that we work together with not only biotechnologists or chemists, but also with economists and people from the management sciences, that we can bridge the gap, especially going from a fuel-based industry towards a bio-based economy. That's our main goal.
Mm-hmm. And I think that speaks very well also to the project that you are leading, that Computomics is a part of, namely the CropML project. I'll say the name for those who aren't familiar with this project. It's new machine learning techniques for more accurate plant breeding by integrating heterogeneous external factors, or CropML for short. This is a BMBF funded project. And I think it ties into what you described as a focus area already, but can you say what's the goal of this project?
Yeah, this is a very interesting project because currently the problem is that the agricultural industry is under great pressure that we deliver or quickly deliver new crop varieties. So we are having changing climate environment. So it's getting more and more challenging that we can grow crops with high yield, and we have to use fewer resources in the future. So we have less space, we need less resources. For example, we are not allowed to use fertilizers anymore, or we should reduce fertilizers. So we have to create and breed new varieties of crops that have still a lot of yield, but become more sustainable and also more productive for also this changing climate environment. So that's the main goal. And we hope that we can help plant breeders and breeding programs by using artificial intelligence and combining artificial intelligence with the traditional work plant breeders are doing. So that's the main idea behind the project. There we collaborate, for example, with Computomics, that we can achieve this goal.
Can you go into a bit more detail? Because you said you're combining the work there that breeders and breeding programs are doing already with machine learning methods, and kind of how does this combination work? Take us through the process.
Yeah, sure. So usually in the last few years, there was a new technique developed. It's called genomic selection, where breeders try to use the genetic data they get from the plants to do predictions on the phenotype, so that we can create more or easier new types of crops, that we can accelerate the breeding process. And the genomic or the classical genomic selection are basic statistical models that look at the genetic variations between the different plants that we have and try to give an estimate about the phenotype of the plant. So, for example, the yield of a plant, or if it's resistant towards a certain disease, for example, or drought tolerance, and so on. And what we try to do is that we use more advanced statistical models, so machine learning models, or maybe also in the future deep learning-based models, that help us to not only look at the genetics data, but that we can also integrate environmental effects, because phenotype of a plant, so how much yield it gives, for example, is not only dependent on the genotype, it's also dependent on the environment where the plant is growing. So we also have to somehow integrate this environmental information, and that's quite challenging because environmental information and all external factors are quite heterogeneous. So we have different types of data. They sometimes are sparse. We don't have all the information for all the different data types available. So we have to integrate this data together with the genetics data. And that's quite challenging. And that we try to achieve by using more modern machine learning-based models, that we can integrate these heterogeneous factors and maybe have then an advantage in the prediction of new phenotypes that then again help breeders again to breed more efficiently.
Mm-hmm. Let's jump a little more into the environmental factors. You said it is quite challenging to get that data, or to maybe even integrate it if you have it. So firstly, what kind of environmental data are you taking into account? Like, if someone doesn't know anything about the process, like, what are huge environmental data sets that you have to take into account?
Yeah, so one factor, of course, is weather. Weather is very important for the growth of specific crops. So we definitely have to integrate climate information, weather information, but also soil information is important. So what type of soil do I have? Which fertilizers have been used before, maybe years before or during the growth of the crop? So these are environmental data, environmental effects that directly affect the growth of a specific crop. So this type of data we try to integrate, and that's so challenging because weather information we can get daily. Climate information we also have. We have historical climate information. We can make a few estimates of maybe how the climate might evolve over the next years. And so it's a lot of additional data that we get. And that's a huge problem that we have here when we try to integrate these environments, because when we also think about the genetics. So if we look at a single plant, then we have several positions in the genome that might differ from other plants. So we have thousands to millions of these positions in the genomes. So we have a lot of data also from the genotype or genetic side, and we have a lot of information from the environments, like daily weather information, soil information, fertilizer information. And to integrate all of this together in one model makes it challenging, because we can't, for example, genotypes thousands to millions of plants. This is a limiting effect that we have in this project. So getting environmental data is easy, but integrating it into the models and combining it with the genetics, that's a huge challenge.
Can you share, or are you already at the point in the project? I believe it started in 2021. So where are you at in the project right now? Are there any results, for example, in the area of integrating the data that you can already share, or maybe give us a teaser for?
Yes, we already have some results, so actually several results already. So what we did in the beginning is we first wanted to get an overview of what is out there on methods that can be used for phenotype prediction and how well do they perform or do they compare next to more modern approaches that are coming from the machine learning side or also from this new deep learning based side, which is just a subdomain of machine learning, but we use neural networks there. And so we compared dozens of these machine learning models with basic genomic selection models to get a feeling of how well these models perform. And this we did on first simulated data. So it's also a challenge to simulate data because they are so complex in this domain. And then we also try to compare it with real data, first in Arabidopsis thaliana. Why Arabidopsis thaliana? So that's a model plant in genetics. It's very well genotyped. We have freely available phenotypes and environments and so on. So it's an easy to get data set, which is very well studied. And then we also used real-world data sets from breeders to evaluate these different models. And this study we just published in Frontiers in Plant Sciences. And there we found out that even though we have now very advanced machine learning models or deep learning-based models, these basic models like basic linear regression, where we add some regularization that can more or less reduce the dimensionality of the data set, they perform still best. So they perform very well on simulated data, but also on real data. And that these more modern models underperform a little bit compared to these basic models or historic models. And a reason for this is that, as I said before, we have so many data points, so many positions in the genome and all this environmental data that it's a challenge to integrate this into these modern methods, because these modern methods have also, we call it a lot of parameters that we have to tune, that we have to find. And that's a huge challenge when we only have a few plants available. And that's the biggest bottleneck that we also investigated. So can we get better results when we increase the number of samples? So if we not only have available 100 plants, but maybe 1,000 or 2,000 or even more, then we see that this neural network-based approaches get better in their predictive performance. But this is also true for the basic models. But we see a trend that the more data we maybe have available in the future, and with more data I mean more samples, the more advantages these modern based methods will have. And that was one study that we investigated, and now we are also investigating in a master's thesis if we can build more or better architecture of these modern based models that can help us to do better predictions on only if we have available, for example, a few samples. And there we see that we can build such architectures. It's getting better. We are not already there that we outperform all the others, but it's getting better. And this gives a lot of potential also for the future research that we can then integrate in these models, also the environmental effects, that we improve the predictions in general. And that was a quite interesting study and also a lot of work, because we had to first collect or build a library that has all these different models that are out there in one common place. And that's what was also done by my PhD students who did a fantastic job there. So Maura and Florian, who also developed here a package that can be used by other academic partners and scientists to investigate phenotype prediction models and to compare them with each other.
Mm-hmm. And how can anyone who'd like to use that access that package?
Yes, that's freely available on GitHub under GPL license, so one can download it. We have also great documentation with this package, with YouTube videos where my PhD students explain how we can use the package, and a lot of tutorials that it's easy to use. So it's based all on Python. So Python is a programming language. So one needs, of course, some Python programming skills, but it can be used as a standalone package, for example, as a Linux command line tool, or it can be also used within a programming environment and easily adapted. And we built it in such a way that we also can integrate new models quite easily.
So you are working on expanding that library, even though it's already a workable and usable package, you're still adding to it as you go along in the CropML project?
Yeah, definitely, because so this is more or less now the basis for all the future research, so that we have a good way to compare different models. And now we have everything that we easily can integrate new and modern methods, and that we also can then compare them again to our old ones. So yes, definitely.
That's quite a range of results already 1 year into the project, or a year and a half, more or less. What would you say are the current main challenges? Are you focusing on the integration aspect more, or which part of the project is currently at the highest priority, if you can share?
So currently the highest priority has that we integrate these different environmental effects efficiently. So this, as I said, will be the next step. And this is, of course, challenging because of all the different things that I said before, that it's so heterogeneous, that we have so many data points, that we increase the dimensionality of the data a lot. So that's challenging, but we already have some ideas how we can accomplish this. And yeah, the next year will be super curious to see what's coming out of these ideas that we have. And yeah, we will see maybe by the end of next year what worked and what not.
And if once you have, so let's, now we're going into the kind of simulation, or into the area of what you hope maybe to get to. But if you manage to reduce that complexity, to integrate that data in a more efficient manner, how do you see the development going from that point?
I think, first of all, if this works out and this will lead to better predictions about phenotypes, then this, of course, will help our breeders a lot, because they can accelerate then their breeding programs a lot and make things possible that might not have been possible before. So they will, first of all, save a lot of time and then also money. And we can maybe then quicker adapt our different plants to this changing and challenging environments that we have in the future, and maybe create then also more sustainable crops. So that's the final goal, of course, and we hope that with these packages that we develop, this will be possible in the future.
Right, and this touches obviously on that very basic question of why it is so important, with where we are at right now with the climate, to integrate those environmental factors into the breeding processes. Because why would you say is it so important?
Yeah, because the problem is we not only have the problem of changing environments, we also have political goals that we have to reduce, for example, fertilizers. So we need also robust crops that need less fertilizers. Or there are also goals, for example, that we are not allowed to use pesticides anymore on the field. So we have to somehow also control the weeds on the fields that are in concurrence more or less with the crops. So which weeds do we have to remove and which weeds can we leave on the field? And if we can create more resistant crops, maybe that can also grow when you have specific weeds next to the crop and they can interact with them, or maybe don't interact with them, that they can get more yield, then this is also a very important thing.
I'd also be interested in do you have a way, or are you attempting to look at location-specific interactions, so the genetic traits of specific plant in a specific region, and to draw maybe from those results to draw into models that would be kind of geographically specific in the future?
Yeah, definitely. I think that's also one of the most important effects that we have to take into account. So usually breeders not only like to create crops that only grow in a specific location. So usually farmers, if they get on the fields and like the crops grow, then they like to get crops that grow everywhere. And if I only have to select first specific crops that only grow in specific locations, then this, of course, limits also the crop in itself. So, of course, investigating the effect of different locations and maybe also altitudes is very important for the general goal of this project, yes.
And then maybe finally on how the project works, you've mentioned at the outset that you like to work in an interdisciplinary fashion and with different partners. How does this principle apply to the CropML project? Who's involved, and why is it maybe beneficial to have the partner structure that you have?
Yeah, so the CropML project would not be possible to do if we wouldn't have strong partners in industry. For example, here Computomics is our main partner in the project. So they are also coordinating the project. And from them, we get, first of all, information about environmental data. They help us to build up the pipelines that we can easily get data from these multiple environments and locations easily to develop our models. So we would not be able to develop new models if we would not get data. And on the other hand, we also have to then prove do our models work. So we have to go back to our partners and our, for example, Computomics, and they have to then go back to their partners to see, can we integrate this more or less into the reality, into our real world, and does this really help? So that's very important. Otherwise, all these developments would be more or less theoretical and wouldn't have any application.
Is there anything else about the project that you haven't shared yet that you find exciting or challenging that you'd like to share with us and the listeners?
That's a very good question. So in general, I think the exciting part is really this strong and difficult discipline of finding models or tuning these more modern-based models towards the effect that we can make it happen, that we can do complex phenotype prediction. What's also something that's very interesting and we didn't talk at all about, so maybe it makes also sense to not only include environmental data and single variant positions from the genomes, but maybe we have to integrate even more. So biology is so more complex than just looking at single positions in the genome. So we also might need to investigate interaction effects between these different locations. We have already information about which genes are involved and are there specific epigenetic factors and so on. So it's a quite multi-omics based approach also. So maybe we can also integrate additional information that help us in the end to create better predictions. But that's, yeah, a lot of stuff that can be investigated, and we have to see how much time do we have for all these different questions, of course, yeah.
I was about to follow up with, is this part of what you're trying to do with CropML in this project phase, or is this almost a teaser for the next project application that the BMBF will have on their desks at some point from you and your partners?
Yeah, I think so. This multi-omics approach won't be part of the project, but this is something that we also can investigate, for example, in master theses. So there are a lot of students also at our campus that are interested in this topic, and they are always searching for challenging and interesting master theses. So we can also open up this box a little bit and investigate these things based on this, on our master students, for example. Yeah.
Cool. Yeah, I think we're making the round now back to teaching, or back to kind of the early stage researchers and how they can contribute to this idea, to this approach. This is very interesting. It's almost hard to summarize all of it, but I think we learned a lot about the CropML project, how you're trying to integrate complex environmental data and genetic traits into machine learning methods to hopefully at some point outperform traditional models on a consistent basis and enable breeders and breeding programs to develop new and more sustainable, more robust plants in a climate that's becoming more and more unstable, and thereby help us all to have a more sustainable model in the future. Dominik, thank you so much for your time, for your enthusiastic depiction of what you're doing with the project, and we look forward to learning more in the future. For our listeners out there, feel free to check out our homepage, computomics.com. We will have a few show notes on this episode with Dominik, and it will have links to the CropML project, of course, and to the website where you can find, I'm sure, links to the package as well that you developed, the library, right, Dominik? Is that
definitely, yes?
Website?
Yes, that's Perfect. On our website and on our GitHub pages. So all information are publicly available.
Perfect. So we will be linking to those. So feel free to surf by there, check it out more in depth, and come back next time for the Computomics podcast. Thank you all for listening.
Thanks for the invitation.